All right, so we're live now, Anna.
Okay, good. So I don't, sorry, I can't see if I can, the slides are being,
Okay, no worries. Then I'll start the presentation and
okay, thank you very much.
you see
So,
the.
thank you. Welcome everybody to this webinar on fibroblast and tissue destruction in arthritic diseases. I'm your host today, and my name is Anne Bay-Jensen. I'm Director of Immunoscience at Nordic Bioscience. I would also like to introduce you to Mayur Bajaz, who will be act as moderator today. But I'm also very excited to be here today with you to discuss this important topic. I'm extremely pleased that our two experts of today, both Dr. Dana Orange, Associate Professor at Clinical Investigation at Rockefeller, and Professor Adam Croft of Translational Rheumatology from University of Birmingham, have accepted to speak to us today. I was in the audience at EULA, and I was super inspired about how the role of fibroblasts in arthritic diseases. And I think this is super important because I don't think it's a cell type we discuss a lot in this field of rheumatology. We normally talk about the different immune cells. So I was inspired by this talk, and I thought it could be fun to bring it in these topics on fibroblasts and arthritic disease into this ECM conference series. So this is the fourth seminar of the Extracellular Matrix Pharmacology Conference Series, and it aims to spread the knowledge on the role of the extracellular matrix in health and disease. Next live conference will be June next year, and so far we have a fantastic lineup. But I personally hope also that we somehow can also include some of the rheumatology field into this, because right now it's very focused on cancer and oncology and fibrotic diseases, and I think fibroblasts have a space in rheumatology in the inflammation. So let's see what we can do with that. So why talk about fibroblasts and tissue destruction in arthritic diseases? So fibroblasts are cells that produce collagen, elastin, and other proteins that make up the connective tissue in the body. Connective tissue, of course, responsible for protecting, for example, the joints. So without the healthy connective tissue, joints will deteriorate. So when fibroblasts are activated in the wrong way, and I don't know what the wrong way is, I hope we will learn something in a short second about that. Then the connective tissue in the joints weaken and break down. This can lead to joint instability, pain, and disability for the patient. So needless to say, the fibroblast activation plays a central role in regulating tissue maintenance and preservation. So, but also said, as I also indicated in the beginning, tissue destruction in arthritic disease is a complex process, and we don't really fully understand that yet. But however, I think our two expert speakers today, Dana and Adam, will have actually make some groundbreaking research to progress the understanding of the origin and mechanism of some of these damages, understanding some of the cell types that are actually involved in some of these damages. And I also strongly believe that such research may also lead to development of new treatment strategies and protect fibroblasts and tissue of the joints, but also other organs. So that was a couple of words from me, but I don't want to take the time from the exciting stuff. So thank you again for joining us, and thank you to Dana and Adam for actually joining us today to enlighten us on the role of fibroblasts in arthritic disease. And I look forward to this productive and informative discussion. So now I will give the floor to Adam and Dana, starting with Adam to give his presentation.
Great, thank you. Good afternoon, everybody. Are you going to put the slides across? Is that...
So Adam, can you see if with the arrow keys you're able to go back and forth if you go under presentation? Yeah. Okay. So one more thing for everyone who have joined, thank you so much. Please put any questions or comments you have in the chat, in the private chat. We will then take some of the questions at the Q&A session at the end of the webinar. So thank you so much, and the words to you.
Great, thank you. So, yep, good afternoon, everybody. So I'm going to focus my talk really around fibroblast activation in inflammatory arthritis and the role fibroblasts play in the pathology of inflammatory arthritis and tissue destruction. And arthritis is an interesting disease to look at from that perspective, because it's both driven by a clear inflammatory process, but tissue destruction is also one of the hallmarks of the disease, resulting in functional disability from joint damage. So the primary site of pathology in the effector phase of inflammatory arthritis is the synovial membrane. So that's the lining of the joint cavity. And what you'll see on the left side is under normal resting conditions, so in the healthy state, this is a very thin lining consisting of synovial fibroblasts, 1 to 2 cell layers thick, that sit on a loose connective tissue. There's no definite basement membrane, it just sits on a very loose connective tissue, mainly associated actually with fatty tissue. You can see that adipose tissue there on the right hand side. And under normal resting conditions, those fibroblasts contribute to the locomotion of the joint by producing lubricin, which lubricates the joint. And also there is a barrier of tissue resident macrophages that sit immediately adjacent to that synovial fibroblast layer, which play an important role in phagocytosing debris from the synovial cavity, which is lubricated by a small amount of fluid. But when you get an inflammatory challenge to the joint, which you can see on the right hand side, you get dense infiltration of inflammatory cells from the bloodstream, you get activation of tissue resident immune cell populations, and you also get proliferation and differentiation of fibroblast populations within this lining, and the lining becomes thickened, and you get organization of a sublining tissue just underneath that, where that loose connective tissue and fatty tissue becomes replaced now with dense infiltration of inflammatory cells and proliferation and differentiation of fibroblast populations. So what you see on the right hand side is that in an established inflammatory insult to the joint, where you have chronic inflammation, you see these formations of subsynovial tissue niches where the fibroblasts become specialized to support the infiltration, retention and survival of inflammatory cells within distinct niches within that sublining tissue. And sublining tissue is always pathogenic. It doesn't occur under normal physiological conditions. It's in response to an inflammatory challenge, or in response to example from trauma. And actually fibroblasts have long been neglected in the kind of inflammatory arthritis field. The focus of all of our current therapeutics are on immune cells, either depletion of immune cells, blockade of signaling, or indeed neutralizing the cytokines produced by those cells. And none of them target directly these fibroblast populations, despite the fact we've known for some time that these cells have a definite role in disease. And some of the best evidence of that comes from the skid mouse model. So when you take human RA synovial fibroblasts isolated from the joints of patients with very established rheumatoid arthritis, you grow them in culture for prolonged periods of time, so they're outside of the joint for a long period of time. And you then implant those cells within a cartilage matrix, a human cartilage matrix, sat within a sponge under the skin of a skid mouse. You see that those fibroblasts continue to invade and degrade the articular cartilage, suggesting there's an epigenetic imprinting that occurs to alter those cells when they're in an inflammatory microenvironment to a persistently activated state, even when you remove them from the joint, and they're capable of continuous destruction to cartilage. So we got interested in, could we potentially target, well, in the initial stage, could we even study synovial tissue fibroblasts that have a pathogenic role in disease, and could we use that as a mechanism to potentially target those cells to alter the disease trajectory or modulate the severity of disease? So at that time, this was well before the sort of single cell era, we were basically looking at what the cancer biologists were doing, and also those in other immune-mediated inflammatory diseases, and looking for markers that were, or proteins that were expressed by fibroblasts at low levels under resting conditions, and then expressed at high levels under inflammatory conditions. And a number of those were identified. We went through a whole panel of screening, but to cut a long story short, we ended up with a protein called fibroblast activation protein, protein alpha, which is a serine protease expressed on the surface of the cell, which is probably important in the joint at attaching the fibroblast to the cartilage. And we looked at this protein and stained for it in patients with synovial tissue biopsies. So we have a pipeline of minimally invasive ultrasound-guided biopsy techniques where we can take small samples of synovial tissue from the lining of the joint at the very earliest phases of disease. And in fact, we can do this longitudinally over the time course as well. And we stain that for this fibroblast activation protein. And what you see in the middle is that under healthy conditions, the FAP alpha is expressed at very low levels. The staining is in brown. Whereas if you then look at patients who have an inflammatory arthritis but go on to resolve, so they don't have rheumatoid arthritis by definition, have other forms of arthritis such as viral arthritis that spontaneously resolves, that the FAP is expressed but at much lower levels than those patients that go on to develop rheumatoid arthritis. So high FAP levels associated with progression to rheumatoid arthritis in a group of patients in which have early inflammatory arthritis. So that led us to the hypothesis that FAP alpha may be a biomarker for a population of pathogenic fibroblasts that have a role in disease. And we knew from the literature that knocking out FAP alpha, so knocking the gene out in the mouse, had no effect on the dynamics of inflammation. It did have a very subtle effect on cartilage damage, but otherwise you saw no difference between the wild type controls. So what we did instead is, thinking about this more of a biomarker of a cell that might be pathogenic, we engineered a FAP DTR mouse in which we coupled the FAP gene with the diphtheria toxin receptor gene. So mice don't express diphtheria toxin receptor. So now with this gene cassette, every time the cell expresses FAP, it also expresses the diphtheria toxin receptor. That means when you give diphtheria toxin to the mouse, you selectively deplete or kill those cells which are expressing FAP. And we've tagged this to a bioluminescent signal. So you can see here that when we induce inflammatory arthritis, you get an increase in the signal of inflammation in the mouse, in the pores of the mouse, seen here in the red. And then when we give the diphtheria toxin receptor, the diphtheria toxin, you see those that are diphtheria toxin negative continue to express FAP, whereas those in which are DTR positive, you see a partial but significant depletion in the FAP expressing fibroblasts. And we've actually done this both systemically and targeted deletion just in the joint, but this is a model called serum transfer-induced arthritis, which we've maintained persistent by giving recurrent doses of serum, so a chronic form of arthritis, and then we've given diphtheria toxin to deplete those fibroblasts. And you can see here that you get significant attenuation on the severity of joint inflammation in the mouse. This is both the front paw and the ankle. And that also associates it with significant protection against joint damage. So you can see here on the DTR positive on bone micro-CT imaging of the mouse during arthritis, if you deplete the FAP-expressing fibroblasts, you get less inflammation, but also less damage. So then it was around the advent of sort of single cell sequencing, and we were clear that not every fibroblast within the joint expressed FAP, and there was a degree of heterogeneity, and we wondered about the full diversity of those fibroblasts within the joint. So we performed single cell transcriptomics of digested synovium from the mouse model using the 10x Chromium platform. Here, this is kind of old money. This is a Tisney plot as opposed to a UMAP that you would use more conventionally now. And you can see that what we found on the single cell transcriptomics, there were 5 distinct populations or 5 distinct clusters of fibroblasts. One of them was an actively proliferating population of fibroblasts. One of them were those lining layer fibroblasts, or they had the phenotype of those lining layer fibroblasts that you see on the joint even under the resting conditions. And then we found 3 populations within the deeper sublining tissue. And the first clue that although that there were not just transcriptomically distinct populations, but there was a functional aspect to that, is when we did the gene enrichment analysis, we found that these populations of fibroblasts differentially expressed genes associated with different disease processes. So those lining layer fibroblasts become activated and they express genes associated with cartilage and bone damage in an activated state, whereas in the deeper sublining tissue, those fibroblasts are associated with immune immune modulation programs, including producing chemokines and cytokines that drive the recruitment and retention of inflammatory cells. And you can see that projected across a pseudotime trajectory analysis here, that you see in the deeper sublining, you see them enriched for genes associated with immunoregulation, as opposed to in the lining layer fibroblasts, where you see them enriched for production of matrix metalloproteinases, for example, which break down cartilage and bone. And we had access to also synovial biopsies from RA patients, and so we were able to perform the same analysis, but importantly then cross-compare those two data sets to show actually that it collapsed down into 3 key populations that were shared in mouse and human, 2 in the sublining tissue and 1 in the lining layer tissue, sharing that transcriptomically distinct function that had a functional significance. And that was important to us because it meant that when we went to interrogate the mice to understand the function of those fibroblasts in disease, it had real clinical relevance to human disease. So here we went back to the mouse. We sort purified those cells within the lining layer and those within the sublining layer. And remember, the distinction was sublining was more immunomodulatory function and lining was more associated with damage and barrier function. And we sort purified and stimulated them ex vivo and looked at what they produced. And you can see that the cells in red, which are these deeper sublining layer populations with an immunomodulatory gene expression program, produce those chemokines which drive the recruitment of immune cells to the joint, and less so in those from the lining layer fibroblasts. But the real proof then came from taking those fibroblasts, adoptively transferring them into the joint of an inflamed mouse, and we were able to show that inflammatory arthritis was more severe and more persistent in those in which you inject those immunomodulatory fibroblasts. So real evidence that they have an immune effector function which is driving inflammatory arthritis, and it was associated with more recruitment of inflammatory cells. So at that stage, we knew that there's certainly an on switch. There's certainly a pro-inflammatory population of fibroblasts that are really doing immunomodulatory function. There's anatomically distinct populations of fibroblasts with non-overlapping effector functions in disease. And in fact, that lining layer population doesn't have any effect on the dynamics or severity of inflammation, but does still drive damage to the joint. But then we were interested in, well, what would be the on switch to potentially switch those immune effector fibroblasts on? And at that time, when we stained for some of those markers, including actually FAP, but CD34 and a protein called Thy1 or CD90, which is expressed by fibroblasts in the sublining tissue, we were able to look at the architecture of the synovium. And here you can see on the outer, these are these lining layer barrier macrophages. But here where you see around endothelium, you almost see this cuffing of a proliferating populations of fibroblasts that are expressing high levels of Thy1 around the blood vessel. And so in work led actually by Kevin Way in Boston, we were talking around this time about these potential mechanisms. Kevin had narrowed this down to potentially a inductive Notch3 signaling, in which you see Notch3 expressed by fibroblasts just around the blood vessel area in the perivascular space, and signaling through arterial endothelium through JAGAD1 or DLL4, you get induction of Notch3 signaling, which switches on a gene program which has a pro-inflammatory function around those fibroblasts around the blood vessels. So the molecular mechanism driving the expansion of those fibroblasts around the blood vessels appears to be Notch3. And Kevin showed that actually if you deleted Notch3 in a genetically knockout mouse, you see a significant attenuation of synovial inflammation and also damage. But also actually if you neutralize that with an antibody, the effect. And in an organoid system, in order to recapitulate those findings, you need endothelial cells present to polarize the fibroblasts into either an immune effector fibroblast or into this more immune effector fibroblast, as opposed to the destructive fibroblast phenotype. So we've been interested in how then fibroblasts regulate the topography of the joint. How do they underpin these formations of these subsynovial tissue niches, and what are the molecular mechanisms that drive that? Because that is a potential therapeutic target in which you could manipulate that process and hopefully impact on chronicity of inflammation. So in work led by Patricia Nissa, one of my PhD students in the lab, she's been using the 10x Visium platform spatial transcriptomics actually to look at the niches within the tissue and define those, and then look at the fibroblasts within those specific niches. And this is just a small part of that data, but then we've looked at what are the fibroblasts doing around immune aggregations as opposed to what they're doing around actually vasculature. And we see that they have very distinct gene expression programs. And it looks like if you're around an aggregate, you're particularly exposed to T cell signaling via interferon gamma, but also other cytokines which induce a particular program in those fibroblasts that are specifically designed to support those cells. And what we find is that there are vascular hubs within the tissue which are active and infiltrate and have infiltrating inflammatory cells. And around those, actually, the extracellular matrix is more loose, and those fibroblasts are producing matrix metalloproteinases that actually loosen up the matrix around those active vascular hubs that allow infiltration to occur. But in vascular hubs that are shut down, where you do no longer see infiltration, actually what you're observing is a much more dense a form of extracellular matrix, and that's driven by matrix producing fibroblasts that actually look very similar to a myofibroblast gene signature, even in the joint, where we don't particularly see fibrosis as what you would classically describe in say the lung or the liver, but you do see extracellular matrix deposition and remodeling, and it appears that that's driven by these fibrotic myofibroblast-like fibroblasts sat around the blood vessels, which tend to work to shut down these vascular hubs. So we were interested in how could we interfere with this molecular mechanism? NOTCH3 is not a great target because it's actually expressed in other tissues. And so we started looking at transcriptional profiling using single cell ATAC sequencing to look at the open chromatin profiles and what were the major transcriptional regulators of this immune effector fibroblast phenotype. And we went back to the mouse model where we can look across the full dynamics of inflammation from the peak of inflammation through to the resolution phase and fully resolved. That's much more difficult to do, of course, in the human setting. And we identified RUNX1 which is classically in the hematopoietic system, but actually appears to be expressed in the mesenchymal fraction here as a key regulator of this immune effector fibroblast population. You can see here this is projected across the pseudotime anatomical trajectory from sublining to lining, and the RUNX1 associates with this sublining phenotype. And we've done some clustering nodes to look at which gene programs RUNX1 activates, and it correlates clearly with this immune effector fibroblast gene expression program. And here you can see that Chris, a postdoc in the lab, has projected that onto the Visium platform, and we can see that in osteoarthritis you see very limited areas of the RUNX1 gene expression program just around the blood vessels actually. But when you look then at rheumatoid arthritis, where you've got chronic inflammation, you see a huge expansion. You see more red here, expansion of the RUNX1 gene expression program being activated in fibroblasts across the tissue. And we think that's a key regulator of this immune effector fibroblast population. And RUNX1 again is not an easy target to go after, but it is potentially therapeutically targeted. And there was a recent paper actually looking at that in lung fibrosis as well, with very similar findings. So going back to the vasculature, it seems to be absolutely key. So remember that I said to you, sublining tissue is always pathological. And it appears that as you get angiogenesis forming in the sublining tissue, that is really sending inductive signals to fibroblasts, which really dictate their phenotype. And the other population, which I'm not really going to touch on, but is a key instructor of fibroblast biology, is the macrophage. And that's a whole other program of work that we're looking at. But it appears that in an activated endothelium, it's giving out key signaling, which is dictating whether that fibroblast is going to be an immune effector fibroblast or actually one that regulates more extracellular matrix remodeling. And RUNX1 is a key transcriptional regulator of that. And it impacts on many disease processes. So that RUNX1 gene program includes IGF-1, for example, and regulating inflammation, but also matrix metalloproteinases associated with damage or remodeling, and those such as CTHRC1 or LERK15, which are associated also with ECM remodeling, migration and destruction. And so here we've looked at RUNX1 and projected this onto spatial imaging and we've looked at where it's activated, and it's mapped here digitally to those which are associated around the perivascular space. You see higher levels of RUNX1. So again, a key transcriptional regulator that probably is partly downstream of NOTCH3 signaling. So I've talked to you about what might be the potential processes that switch fibroblasts on. But clearly, another approach to take is, well, actually, there are many inflammatory episodes that occur in the joint, including in patients with rheumatoid arthritis. Many of them do actually resolve or respond to treatment, or even healthy individuals have transient episodes of inflammation in their joints. So there must be an effective process which occurs to actually switch off fibroblasts back into a normal resting state. And for some reason, that can fail in chronicity of disease. So we got interested in a population of fibroblasts actually were originally identified in the RA synovium, which express high levels of DKK3, which is classically a Wnt antagonist, but has a number of effects even outside of the Wnt pathway. And some of you will be familiar with its effects in cancer, both in extracellular matrix remodeling, but also suppressing local T cell responses. And that's actually also been shown in autoimmune encephalitis models as well. And this is the mouse data here put together, and you can see there's a population expressing DKK3 and LRRK15 actually has a very strong myofibroblast gene signature. And what we found is that when we projected that on a pseudotime trajectory analysis, that as you move towards the resolving phase of inflammation, you see a polarization of fibroblasts towards this DKK3 high LRRK15 population of fibroblasts. So there's huge plasticity here in the fibroblast population, and you see this merge more towards this subset, and we validated that on protein expression as well. So we went back and subsetted at the single cell level, just looking at DKK3 fibroblasts. And you can see you get a number of populations, but the most prominent is this LRRK15 CTHRC1 positive population of fibroblasts. And then we looked at some cell chat analysis to try and predict bioinformatically potential cellular interactions, of which there are many actually, but I've summarized some of the key results here. So firstly, that population of DKK3 fibroblasts express myofibroblast gene signature that we've actually taken from cancer, that we've taken from other immune-mediated inflammatory diseases, and it's very similar to those populations that you see in those other tissues and in the cancer setting. They appear to have a TGF-beta response signature, so they're responding to TGF-beta, and they're inducing that gene expression program. And when we actually looked at what populations might be producing TGF-beta1 and signaling to the fibroblasts, one of the key cellular interactions that came up was actually the macrophage population of fibroblasts, including those tissue-resident populations. Interestingly, some of them appear to be the MERTK population, which is actually sat within the barrier of the lining, which was interesting because on a spatial analysis, that's quite far away, but we think that there may be a countercurrent signaling coming from the lining, which actually is trying to switch off inflammation here and induce an off switch in the fibroblast. So endothelial hubs are really critical for instructive signals, and we also were able to show in an organoid system where we took the fibroblasts out and we grew them with endothelial cells in a matrigel, that the presence of endothelial cells was absolutely critical in order to induce the expression of DKK3 in the fibroblasts. So again, we don't fully understand it yet, but these vascular hubs, what's occurring around that site is either signaling to a more matrix synthesizing population of fibroblasts or a matrix degrading and a pro-inflammatory population of fibroblasts. So key instructions coming from the endothelial cells. And we think that DKK3 really has a key effector role. So this is a working model in progress so far, but macrophages are signaling through TGF-beta, inducing this program in the synovial tissue macrophages, which produce DKK3 as one of their key effector molecules. And DKK3 can both restrain inflammation through altering T cell biology, and we've actually been able to show now that it suppresses T cell function at the level of the tissue, and also induces a pro-reparative macrophage phenotype, and also involved in extracellular matrix remodeling. And you can imagine that that totally fits in terms of you need also repair within a joint after an inflammatory episode, and that repair will involve remodeling the ECM, but also being able to suppress immune responses that are occurring within the tissue. And we've taken DKK3 and actually injected it into the joints of mice, and you can see it promotes a resolution of inflammation compared to the control. And actually, when we look at patients in remission and look at the synovial fibroblasts in remission compared to those with active disease, it's only in the remission patients that you see this high expression of DKK3 in the resting state. So we think even after an inflammatory challenge, having DKK3 expressed by these fibroblasts will be important in maintaining and restoring normal joint homeostasis. And so nearing the end now, but I mentioned before that synovial macrophages give key instructive cues, and we've kind of done some modeling of this in vitro and taken monocyte-derived macrophages and stimulated them with either dexamethasone, which induces a regulatory phenotype in the fibroblasts as opposed to an LPS channel, which drives a pro-inflammatory program. And actually, when we culture those with fibroblasts, they give instructive cues that regulate fibroblast biology. So if you're with a reparative macrophage, that tends to switch off the fibroblast into this resting state, whereas the pro-inflammatory sense signals which drive the activation of those fibroblasts. And so macrophages key instructive cues to the fibroblast. And what we're really interested in there is, why does that cellular checkpoint then fail in chronicity of disease? There must be something that's preventing the fibroblast then responding to the negative regulation from a regulatory macrophage. And that's probably, as of yet unproven, but probably linked to the epigenetics. And when we look at fibroblasts actually present in co-cultured with DEX treated regulatory macrophages, you see they associate with this remission phenotype. So regulatory macrophages switch on this reparative remission phenotype in the fibroblasts. So in summary, then, I've talked about the rationale, I think, for targeting fibroblasts in inflammatory diseases and in inflammatory arthritis. It's an unmet need currently. There may be a number of different approaches to do that, but I've really looked at how you might go for direct depletion of pathogenic fibroblast populations, but that requires good biomarkers and very selective targeting, so you don't have off-target effects in other tissues. Or you look at how you can selectively interfere with the differentiation pathways to prevent those pathogenic fibroblast states from becoming chronic over time. And again, for that, not only do you need to know the molecular biology and have the right targets, but you actually need to capture patients very early in the course of disease. Just like I say thank you to my own team and our collaborators and our funding bodies as well. Thank you very much.
Thanks so much, Adam. Great. So maybe we can take one question in the meanwhile, and then Dana can join on stage as well. So the endothelial cell is of course very interesting. Are there any common denominators between organs if we look at the fibroblast axis and activation?
Yeah, so I didn't touch on that work, but we've contributed some of that work actually being led by Ilya Karunsky at Harvard, actually also with Kevin. And so there we've actually taken both mouse models and then access to four diseases, lung fibrosis, salivary gland with patients with Sjogren's syndrome, where you get tertiary lymphoid structure formation, but also tissue damage to the gland, rheumatoid arthritis, and inflammatory bowel disease, and looked for commonalities. And of course, there are fibroblasts that are distinct to those tissues, but there are also two very common populations of fibroblasts across those. One of them is a vascular interacting fibroblast population, very similar to what I've just described within the joints of the mouse and in the RA synovium. The other one is a T cell interacting population, well, an immune cell interacting population, which is probably more involved in driving infiltration and this immune effector function. I think probably the vascular interacting population, whilst common across those diseases, also if you refine that a little bit deeper, I think it's a little bit more complicated than just one state. So I think it depends on what signal the fibroblast is exposed to first in the vascular space. If that's Notch3 induction, you go down a pro-inflammatory immune effector route. But actually, if you're exposed to TGF-beta and you're around the perivascular space, actually you're going to go down this reparative ECM program. But we don't quite yet know those sort of distinct interactions fully that are occurring. Perivascular space is a very active area with a lot going on. Yeah,
indeed. All right, thank you so much, Adam. Then for those that joined a few minutes later from the start of the session, please put any questions or comments you have in the chat. We'll make sure to take questions at the QA session at the end of the webinar. All right, Dana, then I'll be happy to give the word to you. If you should be able to continue with your arrow keys with the presentation, if you can try that. Perfect. Okay.
Okay. Well, I want to start by thanking the conference organizers for the opportunity to speak to you today. My name is Dana Orange, and I'm here in New York City, and I work at the Hospital for Special Surgery and the Rockefeller University. And I'm a rheumatologist and physician scientist, and I spend most of my time thinking about rheumatoid arthritis. I have this disclosure. So those of you who don't think about rheumatoid arthritis all the time, it's a common chronic inflammatory disease that's most notable for causing inflammation in the joints, but it can affect many different organs in the body, including the lungs, the eye, the skin. And we have quite a few treatment options to manage the symptoms of rheumatoid arthritis, and we can get most patients into remission or low disease activity. But this raises the question, what happens next? Do patients who achieve clinical remission need to stay on lifelong full dose treatment? People who advocate that patients should stay on lifelong full dose therapy would note that almost all of the studies have shown that tapering or discontinuing therapy confers a risk of flare. Sorry, I got a message here. While the advocates of tapering or discontinuing therapy will tell you that not all patients are going to flare, and our drugs are costly and they confer a risk of adverse events. So why expose people to lifelong therapy if you don't really need to? And there's really no consensus from from the professional society guidelines. Like the European League Against Rheumatism suggests that clinicians should do something different than what the American College of Rheumatology suggests physicians should do in the United States. And the Asian Pacific League Against Rheumatism has yet a third set of different guidelines on how to manage this clinical scenario. So what do you do for a given patient? Well, the question comes up, could there be a blood test that might tell you which patients are going to flare? So now I'm going to tell you some of the studies from our group trying to understand what happens when patients flare. And we were really looking to understand what are the specific immune events and what is the order of immune events that happen when a patient develops a flare. We would argue that it's really hard to build a predictive biomarker or a blood test if you don't understand how the system works or what to look for or when to look for it. And so we reasoned that a potentially powerful approach to model and gain insights into changes in rheumatoid arthritis disease activity would be to measure RNA expression over time, using individual patients as their own controls. Now, for longitudinal data to be most useful, the sampling interval should really match the timing of the change of the thing that you're measuring. However, in most clinics, patients with rheumatoid arthritis are seen every 3 months. So here I'm showing you disease activity scores for one patient measured every 3 months over the course of 4 years. And there are little arrows above marking when the patient noted that they were having a flare and called, and a covering rheumatologist gave a prescription for steroids to manage that flare. And when you look at the disease activity scores over time with this every 3-month sampling interval, it's really hard to make sense of the data. You know, sometimes there's an arrow where the disease activity was low and then became high, but sometimes it just looks like there was sustained elevated disease activity. Now, here's what the data looks like when we sample the patient every week with a questionnaire of how the rheumatoid arthritis symptoms are doing. And now you can see that those arrows line up very well with these discrete flares of disease activity. So the problem with studying flares is that flares are unpredictable. And we knew that it would be really impossible to ask a patient to come to the clinic every week for 4 years to collect this kind of data. But this is the kind of data we wanted. We wanted to be able to look at RNA expression data with this kind of granularity rather than this kind of granularity. So we developed a toolkit to enable patients to participate in research from home, where they would respond to the RAPID-3 questionnaire, which is a disease activity instrument to measure rheumatoid arthritis related disease activity. And then they would do a finger stick and put 3 drops of blood into this little microtainer tube that contains an RNA fixative, and then mail that to us each week. And then if they went on to develop a flare, we would go back and look and see what was happening in those samples over time. And the first thing you might say is, you know, how can you think that you're going to measure anything believable from just 3 drops of blood? And so here's how we were validating our fingerstick RNA sequencing by comparing fingerstick RNA sequence data to white blood cell count data that was collected from a regular venipuncture blood draw. And you know, when you take a blood sample from a patient and you get a complete blood count, there's always a differential of how, you know, what % of the cells were neutrophils, lymphocytes, or monocytes. And then we could compare those actual, you know, gold standard clinical blood count data to the inferred distribution of neutrophils, lymphocytes, and monocytes from RNA-seq data, and see that there's a pretty nice correlation. Indicating that RNA-seq from a fingerstick can give you a relatively good idea of what actually is happening in the blood at that moment. And then the next point I want to make is just to give you, you know, highlight the clinical scenario of what it's like for a rheumatoid arthritis patient who is going through a flare. So this is how we're going to look at our samples over time. And, you know, here is the first day that the patient had an increase in her disease activity. And on the back of our RAPID-3 form, we had an area where the patients could free text in, you know, what they said, what was happening during a flare. And so I'm just going to walk you through the clinical scenario of what it's like. And she wrote, yesterday evening, the pain started in my upper left arm and went to my shoulder and then continued to the back of my neck. It was very uncomfortable and warm to the touch. And now her symptoms sort of stay like that. And 2 weeks later, she writes, today I woke up with the right middle finger inflamed and in a curled position. I couldn't straighten it out all day. It was sore and it prevented me from using my right hand all day. I was just feeling blah and I had no energy. So here's what the data looks like when we just look at gene expression data of baseline versus flare. So taking all of these purple samples compared to all of the green samples, for example. And what we found was sort of what you might guess might be increased during a flare of rheumatoid arthritis, because it was a lot of innate immune inflammatory genes, like genes representing monocytes and neutrophils and platelets and things that we know are increased during flare. One thing that did stand out to us were these group of genes that were decreased during flare were things that we're not really used to seeing in the blood, and we didn't really know what to make of that. So then, of course, we didn't collect all of this longitudinal data to look at what's different between the green and the purple, or baseline versus flare. We really wanted to know what is the order of immune events leading up to a flare. And so we managed this analysis by sort of merging all samples on top of each other, relative to the first day of flare, that first time point when the patient said they were flaring. And then we honed in on the 8 weeks before the flare up to 4 weeks after the flare. And we excluded any samples that were from time points where they were treated with steroids, these yellow dots. And when we looked at the data that way, we could see that, relative to the first day of flare, there were these really profound gene expression changes happening in the 2 weeks prior, in these green time points when the patients really had no idea anything was about to happen. So honing in on those, the first, most obvious change in gene expression was this block of red that is increased from the first day of flare and stays increased during the entire course of a flare. And those genes were basically innate immune genes, the same genes that we identified when we just looked at the samples according to baseline or flare. But then we were really interested in these two groups of genes that were increased the 2 weeks prior to flare. And this one was quite surprising because these genes were increased the 1 week prior to flare. And when we did pathway analysis on these genes, they were enriched for things like extracellular matrix organization, tissue development, bone growth, cartilage, and muscle system process. And we don't normally think of blood cells of having anything to do with these processes. So our group had also been working with the Accelerating Medicine Partnership, looking at single-cell RNA-seq data of synovium, and we're aware that there are these subsets of fibroblasts in rheumatoid arthritis synovium. And at the time, things were sort of settled between, two main groups, the lining layer fibroblasts and these sublining fibroblasts that Adam was talking about. So we compared the genes that we saw that were increased the 1 week prior to flare and looked for, was there any significant overlap with the types of cells you see in rheumatoid arthritis synovium? And the genes that were increased in the week prior to flare were really highly overlapping with these sublining fibroblasts. So here's Adam's data that he just walked you through, showing that these sublining fibroblasts are pathogenic in mice. I'm going to skip over this since he covered that so beautifully. And another paper that sort of caught our attention was this paper where they were taking these rheumatoid arthritis or osteoarthritis synovial fibroblasts and transferring them into a mouse and into a cartilage sponge implant. And 60 days later, when they examined a contralateral cartilage implant, they saw that the rheumatoid arthritis fibroblasts had actually trafficked. They had somehow traveled to the other side and invaded the cartilage on the opposite side. This group that was looking at blood samples of patients with rheumatoid arthritis who had either polyarticular disease, so many joints involved, or oligoarticular disease, few joints involved, found that cadherin 11, which is robustly expressed by synovial fibroblasts, by RT-PCR, was increased in the blood of patients with polyarticular arthritis. And when they did flow cytometry to see which cells in the blood express cadherin 11, it was really CD45 negative cells, which are non-hematopoietic cells, suggesting there's a non-hematopoietic cell in the blood of patients with polyarticular arthritis that also expresses cadherin 11, a marker for which is a marker for fibroblasts. So that led us to hypothesize that maybe there's a cell in RA blood that is overlapping with the kinds of cells that are seen in rheumatoid arthritis synovium. And so we used the same strategy that our Accelerating Medicine Partnership consortium used to sort blood sample, synovial samples from patients with rheumatoid arthritis for fibroblasts. So CD31 negative, meaning not endothelial cells, and CD45 negative, meaning not hematopoietic cells, and then potoplanin positive. And we were able to find a population in in the blood that fit those parameters. And though they are very rare, they're more common in patients with rheumatoid arthritis. And when we sorted them and sequenced them, they were expressing the same genes that were increased in the blood just prior to rheumatoid arthritis flare. And another group made a similar observation. They were trying to identify whether they could predict which patients who were in remission were going to flare when they discontinued their therapy. And they were looking at cytokines in the blood. And one of the cytokines that was the most predictive of flare compared to sustained remission was this IL-34, which is also robustly expressed by rheumatoid arthritis fibroblasts. So this led us to propose this model of the immune events that lead to rheumatoid arthritis flare. And I sort of glossed over the gene expression signature that was that we saw 2 weeks prior to flare, which was really an activated naive B cell signature. And then 1 week prior to flare, we see this fibroblast-like signature. But amongst ourselves in the lab, we felt so strange talking about a fibroblast in blood that we didn't really want to call it that. We kept calling it the mesenchymal cell because it doesn't express CD45. So we came up with the moniker prime cell for pre-inflammatory mesenchymal cell, and that we see this signature increased in the blood prior to flare. And then during flare, the gene expression signature really goes away. And so we hypothesize, we don't know, that it could traffic to the joint and potentially differentiate further and play a role in inflammation. Though, of course, there are other possibilities. It could be dying or they could be going somewhere else. Those are things that still need to be figured out. And with that, I thank you all for your attention, and thank the collaborators who participated in this work. It's really a group project, and I also want to thank the patients who were incredibly diligent in sending in their blood samples and their disease activity scores every week for a very long time.
Great, Jonah. Thank you so much for your presentation. One quick question. Do you think prime cells are contagious?
Contagious meaning from one person with rheumatoid arthritis to another?
Maybe I can, because I had similar questions. So I think what you mentioned that you could find, that some showed that the cells were found in come from one joint and they could track it to another joint. So contagious in the sense of moving within a person and then contaminating, if you will, another joint.
Yeah, I'm very interested in that question. I don't know. The reason to think they might be contagious is that, all of the flares that we sequenced were really polyarticular, and they were, as I described clinically, like they were going all over the body. It wasn't like, somebody developed arthritis in their wrist and that was the problem the whole time. Like it was additive, and even some joints could come and go over time. So that is sort of intriguing to me, that clinical phenotype. And of course, there is a precedent in the mouse that synovial fibroblasts can traffic. Reason to think that they're not is that the gene expression signature that we see in the blood, it's synovial fibroblast-like, but it's a little bit more primitive and almost like stem cell-like. When we do flow cytometry and you backgate and look for forward scatter, side scatter, they're very small. So, and fibroblasts are large, so it's hard to imagine that a big fibroblast like sort of condenses itself and then goes traveling in the blood. So for that reason, I kind of think that maybe they're like a progenitor cell or like the precursor of a tissue fibroblast, but I'm not sure.
Science needs to show, I guess.
Yeah.
Great. Thank you so much. All right, then I'll give the word to Anna. Anna, please go ahead, and you should also be able to use the arrow keys to continue.
Yes, thank you very much. And it's very difficult to follow Dana and Adam on this, but I will try at least because we kind of, my group and I see rheumatoid arthritis from another angle, although we lean on the science by experts and expert scientists like Adam and Dana. We also try to see if we can measure differences in patients. So I will give a small story, first a bit of background on identification of fibrotic and fibrolytic endotypes in rheumatic disease cohorts, a study that we did together with Janssen to try to understand if we can identify endotypes by measuring something in blood. I should mention that I have a disclosure. I'm a full-time employee and shareholder of Nordic Bioscience. So what I'm mainly interested in is actually the extracellular matrix and how that is remodeled as cause of disease, but also as cause of repair, for example, as treatment response. So there are many different collagens, there's 28 of them, and you can find many of those in joint tissues, actually, both in cartilage and bone, and in the ligaments, in the synovial membrane. So it's a mismatch of different collagens. We typically talk about type 1, 2, 3, 4, the type of collagens we're interested in, but we are basically interested in many of these collagen and how they are degraded as part of disease. So just to give you an idea how we see the world, so when inflammation hits cells, they become activated, start releasing MAPs and catechisins, elastases, and other proteolytic enzymes that then start remodel, degrade, and destroy the collagen or the protoglycans, so other extracellular matrix molecules in the extracellular matrix in the tissue, including also the joint tissue. And of course, some of these small fragments can be picked up in blood and thereby reflect this remodeling. And that's the focus of my research group. So just to give you an example of type 2 collagen, and I know other people have other favorite collagens, but mine is type 2 because I worked with that for 2 decades. And if we look at the type 2 collagen molecule, it's actually by remodeling and destruction destroyed in different pieces, and each piece tell a different pathological story. So for example, CTX2 we have here, that's a measure of reflection of cartilage destruction, and it's measured in urine. There are other cartilage destruction. So this is an atelopeptide here we have in the interhelical region, and it reflects the full destruction of type 2 collagen. But if you look at, for example, the propeptides in the end here, if we can pick up fragments of the propeptides, it's most likely a reflection of cartilage formation or type 2 collagen formation. So we can use these different signals to kind of understand what's going on in the tissue as part of the pathology, but also as part of the treatment response and understanding what treatment does to the tissue. So we have been working a bit for the last couple of years thinking of how can we understand the patient using measuring some of these collagen fragments in blood. So if we look at one axis here, we have fibrosis biomarkers, so all the pro-collagens, how if we measure those, what do they measure some kind of fibrosis. And the degradation fragment, they measure tissue destruction in some way. And what we see now when we look across different fibroinflammatory disease and rheumatology diseases, that a large proportion of diseases have a high signature of destruction. So they have a high level of type 1 collagen, type 2 collagen, type 3 collagen degradation. Whereas other diseases, for example, in the fibrosis field, if we talk about liver fibrosis or lung fibrosis, they have a fibrotic signal, but low destruction signal. So you could say these are the clean fibrotic diseases, and here we have the decorative diseases. And then there are the high turnover where there is both a fibrotic and a fibrolytic component in disease. And we even have diseases like osteoarthritis that seems to have low turnover disease. So, but then of course this is a schematics and the world never looks like that. So we kind of try to understand how these may overlap and interplay. But before I continue with that story, I just want to give you a bit of background on some of the biomarkers here. So if we, for example, look at type 3 collagen, and that's the theme I want to focus on, because there is a connection to fibrosis in this. So type 3 collagen degraded by MMP9. We see that this in RA patients are correlated to disease activity. And we also see that it's in different studies. We also see that a biomarker like type 4 collagen degradation, so that's mainly basement membrane, where type 3 is mainly interstitial matrix degradation. We see that there is also a signature towards being association with erosion and sharp skull, so bone changes, these joint structural changes in here. So I think that's important to know, that this is not just that tissue ECM remodeling is actually associated with how patient functional feels, but also the pathology we tend to measure in the patient-reported outcomes. If we then look at, for example, treatment response, so we have methotrexate here. If we look at the right panel and look at type 3 collagen, the blue here that's placebo-treated patients. The purple in the middle here is methotrexate-treated patients, and then we have tocilizumab-treated patients. And this is from the Ambition study that was recently published. And what we saw in this study was that if we look at type 3 collagen destruction, we can actually see that we can modulate that, especially with an anti-IL-6. And this makes sense because an anti-IL-6 goes in and interfere with the production of MMPs from some of the mononuclear cells. So this makes sense, but the effect was smaller, of course, in the methotrexate patient. And of course, we did not see anything in the placebo patients as compared to the others. But it's just highlighting there is a connection between what we try to modulate and some of these biomarkers that reflect tissue turnover. So we try to profile that. And also, just to give another example, if we take C3M again to be consistent, we can also see in this study that methotrexate was compared to adalimumab, to tocilizumab, and to tofacitinib, so different pathways. And what we saw was that the greatest effect was seen exactly with the anti-IL-6 and the JAK inhibitor here. But there was another interesting thing I want to highlight here, which is, as in the beginning in the introduction also mentioned, was that there is a fibrotic component in some of these. I mean, generally we think RA is a one, at least from our perspective, a very destructive disease. We see high tissue-related collagen destruction biomarkers, but we also see that a subset of patients have this PRO-C3, so type 3 collagen formation high profile. And what we learned from that was that there is also, and here we start seeing really differences whether how well a drug modulate that part. So for example, here it was only the JAK inhibitor that could lower the level of fibrosis. That could also indicate that the potentially IL-6 would not work, anti-IL-6 or TOSI would potentially not work in fibrotic related diseases. But of course, it's not up to us, but it's a nice way to see that we can actually, with measuring what's going on in the tissue, we can actually get an idea of how the different drugs affect these tissues. So it's a proxy of doing a biopsy, for example. And also we looked into different studies and look at the level of some of these biomarkers. We could also see there was a relationship with the structural outcomes like sharp score and others, and see whether there was some reduction in the biomarker or in the tissue destruction biomarker, whether that was related to the primary outcome in some of these studies. And we could see that we need to significantly reduce the level of tissue destruction to actually see a positive outcome for some of these drugs. Otherwise, potentially, it is not good enough. So tissue is important in RA. I think that's the message I want, and important in other inflammatory diseases, including other rheumatic diseases. We need to modulate the tissue, not only the immune cell pathway, but also the tissue that's really being affected by the disease. So let me go back to where I started. So So what we observed is that there are these chronic imbalances between fibrosis and fibrolysis in fibroinflammatory diseases and also in rheumatic diseases. So what we did was that we wanted to look at different endotypes, or try to understand, are these, I mean, although patients with distinct diagnosis exist, can we, by looking at these tissue outcomes, these blood tissue outcomes, can we then identify different subtypes that we would not identify by just looking at the patient or assessing the patients? So the idea was to include a lot of patients, look at the data, try to do clustering and see what do we get out of that. So this is kind of a study that should prep the field and get us to think about how we can look at the patient in a different way. So hopefully some will get inspired, and I would love to talk about how to progress in the future. But what we did was to measure this rheumatoid panel plus some fibrosis biomarkers in the placebo group of 4 different studies together with Janssen. So my co-authors are listed here. And we did hierarchical clustering. So this included the rheumatoid panel, but this included healthy individuals, AS patient, PSA patient, and lupus patients in the end. Yeah. They all had active moderate to severe disease level. So what we did first was just to look at the levels of tissue destruction biomarkers. So type 1 collagen destruction, type 2 collagen destruction, type 3 collagen destruction, type 4 collagen, type 6 collagen, and type 10 collagen destruction. So we did see some, I mean, on a general look, it actually looks like most of these destruction biomarker are elevated in diseases, but we also see that diseases that we have not been successful in profiling a lot, the lupus patient, for example, we also see similar some common denominators with diseases like AS and PSA. Surprisingly, we did not see anything in the C2M, but we had a really nice signal with C3M. And one of the jokers in the study was that we wanted to look at type 10 collagen because that's associated with endochondral bone formation. And that's something that I think is especially important for AS and PSA patients. So, and here we actually see a significant elevation across the different diseases as compared to healthy controls, especially PSA and lupus patients. But we also, which is I think rather new thing, also looked at the fibrosis biomarkers, so type 1 collagen, P1 and P, not so many surprises, type 2 collagen formation. But what I think is one of the more interesting one is ProC3. So we know that ProC3 is being developed as a diagnostic tool to find fibrotic patients in, for example, liver fibrosis. So this is a biomarker where we can identify patients with the fibrosis, and we wanted to see whether there was patients here in these populations that we not normally characterize as fibrotic patients that have these signals. And what we find was indeed that especially PSA patient, but also lupus patient, had elevated elevated levels of this fibrotic biomarker ProC3. And I think that's super interesting because that adds a different layer of complexity and understanding of some of what is the driver underlying drivers of some of these diseases. So the next thing we did was to do hierarchical clustering, and what we see here is that that when we do that, we decided to end up with 4 different clusters. And the first cluster here, the gray inside here, that's the healthy controls, and the red around here, that's the patient that fall into the cluster 1 that resembles the healthy controls. And then in cluster 2 here, what we see is, if we look at the list over here, what we can see is that the biomarker that comes up is VCAM and collagen type 10. And I said before that type 10 collagen was a bit of a joker because we don't understand that, but it's super interesting that one cluster is actually driven by that type 10 that bonal endochondral formation, turnover type. And I think that that's a note for the future, because maybe there is a subtype here where there is a more bonal target for that. Then, so that's why we propose this to be endochondral endotype. Of course, this is proposals because we just have one study here. So tissue destruction endotype, that's again a proposal, and we call it that because what we see here is that type 6 collagen and interstitial matrix turnover marker is elevated in this, and also the Wickham biomarker, which is the citronated and MMP degraded fragment of vimentin. So it's not really a tissue biomarker, but it's associated with that interaction with immune cell and fibroblast for it to be released into circulation. And this last cluster here, that's the cluster we call fibroinflammatory endotype. And what we see here is that many of the basement membrane interstitial matrix turnover biomarkers, both the fibrosis and the fibrolytic one, are elevated in these patients. So then comes, how does that relate to the clinical outcomes then? I mean, and the first thing is that it's not that many big associations. So it's not that we would have been able to identify these endotype or these pathological endotypes by just looking at disease activity scores, because that's not what's enough. We see some association with the CRP across the board. There is a bit with ASDAS in this across the board, but also with erosion scores in PSA patient and a bit with BMI and SLE patient. But what is interesting here is that here we have a picture of something that's independent on what we can find by looking at the patient-reported outcome and the clinical scoring. And I think that is a way of exploring some more, because this may tell us where should we target. And also that when we look at the distribution of patients, we can see that the PSA patient and the AS patient fall mainly in the last two clusters, while the SLE patient, the lupus patient, that we have a huge problem in differentiating falls into the cluster 2 and cluster 3, where cluster 2 was the one with type 10 collagen regulation up. So just in summary, to close this off, then tissue destruction and fibrosis play an important role in arthritic diseases, and we can use biomarkers of tissue destruction and fibrosis to potentially understand what's going on in the patient on a given day in a given situation, and also potentially drive the decision making next to other technologies that were presented by Dana and Adam to find new efficacious treatment. So one thing is to find the target, the other one is to judge whether it would be efficacious and target the tissue that we want to repair and sustain. So and also that tissue biomarkers may provide these tools for identification of treatable and druggable endotype that helps us differentiate this heterogeneous formation. And then at the end, I would like to thank my team and, of course, my collaborator at Janssen for allowing us to present these data that hopefully will be published soon. But I think I would like to spur that discussion on trying to endotype using measurements of tissue destruction and fibrosis. So thank you very much for your attention.
Thank you so much, Anna, for your presentation. Great. Then we've arrived at the Q&A session. So I would like to invite our speakers back to the stage.
Hi.
All right. Let's see if we can get Adam. Oh yeah, there we go. Great. Thank you so much. So we've received a good amount of brilliant questions over the period of this webinar, and then hope you can answer those with your expertise. For the audience, if you still have any questions, please put them in the chat, and we will make sure to use and ask them. So let me close the presentation so we can see the attendees. So let's start with this question. Adam, as you establish FLS cultures for co-culture as assay, are there any concerns that FLS grown on plastic may no longer be physiologically relevant, and that could be due to changes in transcriptomics, etc.
Yeah, that's a great question, actually. So there's a couple of things. So actually, if you do take them out of the joint from very early disease and you grow them in culture, actually they do tend to lose phenotype. We've assessed that actually, and Kevin's published on that as well, which is single cell transcriptomics, that they enter this kind of null phenotype. They all look the same. You have to kind of polarize them with some stimulus, whether that's endothelial cells, which will stimulate the immune effector, or you stimulate them back with cytokines. Actually, what's quite interesting is if you stimulate them back with cytokines, they do tend to have some memory. Now, that may be different if you go to later disease, really established disease. There, when you, and as I alluded to on that data with the skid mouse, there's a huge historic amount of data that shows that if you take fibroblasts out from really established disease, they have this persistently activated phenotype, which is maintained actually in culture. So in other words, if you early disease, probably more plastic, inducible phenotype that's highly plastic. Maybe in later disease there's a polarization that's epigenetically mediated that maintains them in a certain phenotype. But it's definitely a concern in tissue culture plastic that they will enter a kind of null phenotype. They need a further stimulus.
Thank you, Adam. All right, Dana, are prime cells identifiable in mouse models of RA? And if so, have you done any lineage tracing studies where they may end up?
We don't know, but we are looking at that now. We're very interested in that question. Yeah.
Thank you. And then a follow-up question: the stem cells, are they bone marrow derived that you talked about in your presentation?
We don't know where they come from. They're not hematopoietic because they don't express the CD45 marker. But we have big debates about where they come from, whether they come from joints or whether they're attached to endothelial cells in the vasculature and they sort of dislodge, or whether they come from the bone marrow. We're trying to figure that out.
Thank you. Adam, do you think today's treatments could have a negative effect on fibroblast phenotype and chronicity?
Yeah, so I think it's all about the timing that you go. So one of the things we're quite interested in looking at is not only sort of snapshots of fibroblast biology, but how that changes across the natural history of the disease. So I think if you target these, even if you neutralize certain immune-driven pathways at the late stage of disease when the fibroblasts are still have quite a plastic phenotype, then I think there's a chance that you can modulate their biology. But if it comes too late and that synovial microenvironment is already persistently altered, the fibroblasts are doing the predominant driving of disease. And I think, as Anne alluded to, a lot of this will be about tissue pathotype as well. I mean, the pulse immune pathotype is really interesting because you have low immune cell infiltration, and you have dominated by sort of collagen or matrix producing fibroblasts, but yet don't forget the patient has active disease, sometimes in the joint. That's why we biopsied it. So it's kind of strange what's driving that. It's not entirely clear actually. And there the pathways might be very different. So I think it's a bit about selecting the right endotype, pathotype of the patient, but also stage of disease.
Great. We also have a couple of questions just aimed at you generally. What makes the right therapeutically viable area of the RA joint to target? Is it the fibroblasts or inflammatory cells or vascular cells? Or do you recommend a combinational therapy at various stages?
Do you want me to go first? So I think that it's going to be combinational. I think that in the end, the right kind of fibroblast targeting therapy with an immune depleting or immune targeting treatment could be highly effective as an adjuvant, particularly in those with refractory disease. But it's also about getting much better at precision medicine, as my other two colleagues have alluded to as well. So more about stratification to the right therapy, whether this is fibroblast driven or largely immune driven. It's clear that the lymphomyeloid pathotype is probably very highly immune, and you're going to see some benefit from targeting immune. So I think it's it's precision stratification to get the treatment right, but it's also about thinking about using therapies in combination, I think.
Yeah.
Dana, Anna, go ahead. Sorry, Dana.
So as I said, we have a lot of great treatments. We can get most patients to remission or low disease activity, but none of our treatments are curative. So that tells me that there's something else that is missing that we're not treating when we use, the agents that we have at hand now. And so I agree. I think probably there's, probably a fibroblast that is pathogenic that needs to be targeted in addition to our regular anti-inflammatory approaches.
Anna, if you have anything to add.
No, I think both Adam and Dana provided the best.
Thank you. Then a question for you, Anna. Let's see, is fibroblast activity related to the response to anti-inflammatory therapies?
Is this to me, sorry, or was this on?
This is for Anna.
But I guess also Adam can answer.
But Adam can answer.
Because I think Adam knows better than I. But I mean that's a good question, and I don't think no one looked at that at the end of the day, right? At the moment, not really. So that could be a target to target the fibroblast in disease. So I will let Adam answer because he's much more knowledgeable about the fibroblast responses.
Well, I think it's, I agree, and I don't think it's known yet, but I think it is coming actually. So I think that kind of data will be around once we start doing bigger meta-analyses of single cell data. We'll get a finer grain on which fibroblast subsets associate which potential treatment responses. And the cancer biologists are already doing this, right? So because they get access to tissue, they're already stratifying, not just on the basis of whether you just express one receptor or not, but they're going to fine grain analysis on the single cell and looking at the subsets of CAFs that are involved in stratifying chemotherapy on that, certainly in a trial and research setting anyway. So I envisage that's exactly what, whether it's a fibroblast or a macrophage or T cell or whatever, that's the kind of resolution to the biology we're going to be looking at in the future.
Yeah, I'm just going to add to that, to what Adam was saying. In the Accelerating Medicine Partnership Consortium data, those low inflammatory, POSI immune or fibroblast-rich samples, they tended to come from synovectomy, because in order to make it through the single cell pipeline, they needed enough cells, and those samples don't tend to be, full of inflammatory cells, or they're not so densely packed with cells. But the fact that they came from synovectomy means that the patient, the rheumatologist, and the orthopedic surgeon all were so impressed with that joint that they decided to take him to surgery and remove every bit of synovium that they could. So to me, that says that we're really missing something in our clinical management of rheumatoid arthritis. It's not all about the hematopoietic cells. There's something else in there that needs to be targeted.
Okay, maybe also I can have a follow-up question, maybe both to you and Adam. It's about, is there a kind of you can say a balance between good and bad fibroblasts? Do you think in RA? So I know in some of the other fields that I follow a bit, we talk about destructive fibroblasts, so it's named different in different fields, but also repairing fibroblasts that repair wound healing, part of the wound healing scarring process. So how do you see the world with that balance between the evil and the good?
Yeah, so it's a really good question. So I think at the start of this process, when we first started getting this type of data, I thought that it was going to be about composition of fibroblasts within a joint, or and that's why you see susceptibility of certain diseases and certain patterns of joints, and maybe there's an element of that and that kind of data is also coming where we'll have comparisons between why is the certain finger joints affected in RA and it doesn't necessarily affect others, but osteoarthritis affects all of them. So I think composition is important, but I actually think they're much more highly plastic than just sitting in just subsets that don't move. So I think a lot of it's about the intercellular signaling that occurs and the tissue niche that develops over time. So I think it's more complicated than just good and bad, and it's a pretty great. I think it's how the real question is how does it actually evolve over time? I think like Dana said about flare. Why is it you can have somebody that's actually treated effectively with an immune targeted therapy doing okay, and suddenly experiences a flare and gets breakthrough disease that then doesn't settle? I mean, what's the kind of immunology driving that? And I suspect that's a complex interplay between the immune and the fibroblast that changes the actual microenvironment. Yeah.
Thank you. Dana, can you discuss the potential therapeutic interventions that would utilize the knowledge from prime cells to alleviate RA symptoms?
Well, I think we have to work out whether prime cells are pathogenic or not. I still don't really know whether they're actually like a wound healing response or whether they're pathological. And, of course, then if we knew that, we could either help everybody have more prime cells if they're good and they actually help, heal the wound of that, of inflammation in the joint, or deplete them if they're pathogenic.
Right. Adam, if you culture fibroblasts in stiff cell culture dishes, is there any possibility to transform fibroblasts to myofibroblasts because of the stiffness?
Yeah, so I don't know the answer to that, other than we've been, and maybe actually somebody that has more myofibroblast knowledge than me will know, but we've struggled because that population, if you try to bring them out of the joint, we don't get a huge amount of success. We don't get huge numbers, probably a few more than Dana gets of prime cells from the blood, but it's a difficult population to work with. So we have looked at ways of stimulating that phenotype. I mean, TGF-beta looks like it does it largely, but not entirely matches up to exactly to the phenotype that we see in the joint. So it might be interesting to try and put them in sort of collagens and things like that to see if we get a stimulus. It's a good idea, but we haven't tried it actually. Yeah.
Thank you. And I think this question is for Dana. What role could the microbiome play in flare-ups?
Well, we have a whole paper that I didn't talk about this data, where we were looking at that question. We wondered, could there be some microbial trigger to flare? And I'll make a long story short that we sort of stumbled into in our longitudinal samples that there were oral bacteria getting into the blood in some patients. And those patients happened to have severe known periodontal disease, and they were getting multiple treatments for their periodontal, like teeth extractions, and they were on and off antibiotics for that. And we could see that when they were having oral bacteria in the blood, that was associated with an inflammatory monocyte response that is also seen in the joints of patients with treatment refractory rheumatoid arthritis. So that was very interesting to us, because it's known that patients with rheumatoid arthritis are more likely to have periodontal disease than those without rheumatoid arthritis. And if they have periodontal disease at that moment, they are more likely to be refractory to treatment. But that's like a whole other story that anyone can look up. That's published in Science Translational Medicine this year.
Thank you. A follow-up question: does prime cells transcriptome switch closer to that of a mesenchymal stem cell as they enter the bloodstream?
Well, we don't have that kind of like positional data on them. We only have them when they were in the bloodstream. So I don't know about that question. I can't answer that question.
Thank you. Let's see, one more question, Anna. What would be an ideal biomarker of fibrosis, or what would be ideal fibrotic endotype? Should it correlate to patient-reported outcomes, to treatment response, or what do you think is the holy grail?
Yeah, I think, not so much in RA, but in osteoarthritis, we've done quite a lot of studies on for 20 years, trying to correlate some biomarkers with the patient-reported outcomes and imaging. But I think at this stage, I actually think we need to separate things into different boxes. So I think some of the biomarkers that we are interested in, but also other biomarkers, they're a reflection of disease activity rather than disease stage or disease progression. So I think we need to think of the molecular signal that we measure in blood more as things that is complementary to what we ask the patient of, so the patient-reported outcome and how they function and feel, but also how we look at the imaging. I think it's 3 different complementary assessments to understand the patient, and so I don't think we necessarily need to correlate to any biomarker needs to correlate to pain. It's nice because it links pathology, but I also think it's dangerous because what we want to know in some of these diseases is more what's going on, so we can target that. We want to know when they have flares and what type of flare they have, not necessarily something that replaces how patient function or feels. So I don't know if that answered the question, but I really think that it's 3. We need to think of it as 3 different things that gives us different information.
Thank you, Adam. Dana, do you would like to add anything to that?
No, I was just agreeing. It's
Yeah.
I agree. I'll add that when we looked at our flares, we could see there is some heterogeneity in flares. Not all flares are exactly the same. And, we're trying to figure out what that means.
Right. Thank you so much. There was one more question, if we could share again the information about the ECM conference next year. So let's see if I put this. Anna, could you just slightly mention again about the ECM conference next year?
Yeah, just to say that this, of course, is seminar series is part of, it was the 4th webinar on trying to link fibrosis and ECM remodeling to different diseases. And of course, there is a live event in Copenhagen next summer, where there's a really nice lineup of people that's expert in the field of understanding the role of the extracellular matrix in disease, but also as part of treatment target and pharmacology. And of course, I'm personally hoping that we also at that can have a parallel session on what this means in arthritis and immunology, but that needs to come. But right now, I just want to thank also the speakers.
Yes, thank you. We will make sure to,
it was very educating, and I hope that we can expand the knowledge about fibroblasts and the fibroblast phenotypes in arthritis and immunology in general.
Absolutely. All right. I would also like to thank the speakers and for your invaluable insights, and I'd like to thank the audience, of course, for joining our sessions. And I hope we expanded your knowledge on the ECM and fibroblasts in arthritic diseases. And we hope that you'll join the next session as well, because that will be focused around lung diseases. So we're excited to organize that soon as well. Thank you all, and have a wonderful day.